Hi everyone, I'm very happy to be here to introduce my work to you. My work is about the comparative genome analysis of immunomodulatory ability in these two species from Japanese pickles. So here is the overall view of this work. I will introduce the details later. And what is the novelty of this work is that we compiled the CH index, the Calinski Harabasz index in this work for the comparative genome analysis. So why we want to use this index to develop this novel pipeline? First of all, I'd like to introduce the background of this research. Uh, we know that the lactic acid bacteria can provide the aroma, flavor, health effects, and the shelf life. And they are from diverse resources, which is closely related to our common life. And they can also manage the gastrointestinal disorder, and they can also provide the immunomodulation in our immune systems. So during the immunomodulation, they will induce diverse immune responses, which can produce diverse cytokines. Of these cytokines, the interleukin 10 and 12 are drawing the attention from researchers, since they are both of them are playing opposing roles in our immune system to keep the homeostasis. And of all these lactic acid bacteria, the Lactiplantibacillus, plantarum, and the pentosus are drawing attention from researchers, since in their genomes, the previous reports found that they conserve diverse probiotic marker genes. These genes are associated with adhesion ability, gut persistence, and stress resistance. So on lab, the MMP is expressed on immune cells. The pattern recognition receptors is expressed. So during the interaction, the immune responses will be induced. However, there are gaps of these researchers. The probiotic marker genes only suggested the immunomodulatory capacities, while the genomic exploration did not identify the genes directly related to MAMPs on bacterial cell membrane. Therefore, we have the aims of this work. The first one is to find the immunomodulatory of plantarum and pentosus strains originated from Japanese pickles. We collected the samples from all these four prefectures in Japan, and we curated their genomes, and we examined their cytokine productions via ELISA measurements. Next, we want to discover the direct immunomodulation linked genes of these strains by a novel comparative genome analysis pipeline. In this analysis, we compiled the CH index in our research. We used the genetic distances from the orthologous gene groups from their constructed pine genomes, and then we corrected this CH index to PG index in this work, and then we recalculated the CH index again based on k-means clustering analysis. We used these two index for the screening analysis to screen out the candidates, and we extracted the amino acid sequences of the candidates for a functional annotation. Then we figure out the final potential one. Here are the results. Before the comparative analysis, we found two clades. So one clade is called the interleukin 10 clade, and the other clade is the interleukin 12 clade. These cytokine production differences is parallel to their genomic differences. This is the results for plantarum, and we also observed the similar differences, the parallel differences in pentosus. So this is the KY4 interleukin 12 inducing clade, and this is the KY5 interleukin 10 inducing clade. So based on these differences, we perform the comparative genome analysis, and then we screen the candidates out. For plantarum, it is the group 1590. For pentosus, they are 728 and SEC-A. After the gene content exploration, we figured out that these two group genes were encoded tag F, and this tag F is responsible for the poly-GroP WTA biosynthesis. So this cell wall component is a potential MAMP to induce the immune responses. From previous reports, we know that these genes are responsible for the poly-GroP WTA biosynthesis, and furthermore, the researcher found that in WCFS1, the plantarum, the tag locus is involved in the poly-GroP WTA biosynthesis, since there are size differences were observed between one TAGF and the second TAGF, and this one might act as the GROP primus, the tag B. In our research, we also found the size differences, and we named the group 1590 and the 728 as TAGF2. So other WTA biosynthesis related genes were also distributed across the TAGF2 conserving strains. Therefore, we applied this KY4L3, the plantarum, for a mouse model construction. And from this mouse model construction, we confirmed the interleukin 10 producing ability of this plantarum strain. Therefore, we postulated this schematic plot of the poly-GroP WTA biosynthesis pathway in these TAGF2 conserving strains. Here it is. Here comes to the conclusions and the visions. So the detection of this TAGF2 in both species confirmed the effective use of our comparative genome analysis pipeline, and we are applying this pipeline on a larger scale of datasets. So we're also applying these immunomodulation active strains found in this study to experiment it for their therapeutic potentialities on mouse model. I'm here acknowledging all the contribution from all the collaborators and the supporting from my colleagues. Thank you for your listening.